Okay, hi everyone. So my name is Charlotte Coates. I'm a master's student candidate at the University of Guelph, and I've been working with Dr. Peter Kevin for the past 2 years on this project before I started my master's, but I'm very happy to be able to do my master's now around this project as well. So to introduce, our topic is the temperatures within horticultural plants, specifically stems and flowers. Now, we are aiming to explain rapid growth by micrometeorology, morphology, and physiology. And our project is, of course, sponsored by COHA and the AAFC, so we are very thankful for those funding support, and we are at the School of Environmental Sciences at the University of Guelph. So firstly, I'd like to introduce you to our project, which is on a new area of science, which is called endomicrometeorology. And micrometeorology around plants is well studied. Of course, this refers to the climate near the ground, and micrometeorology has been used in vast applications in agriculture already. So we know that the atmospheric movements of wind and convection of air and light conditions and radiation, the temperature and heat in the field, humidity and moisture in the air, and the precipitation as well. These aspects of meteorology are all well studied in agricultural systems, and we're seeing the trend towards moving towards precision agriculture to increase the accuracy of being able to study these, increase the accuracy of the data collection of these different parameters. Our project actually aims to answer the question of what micrometeorological conditions exist within plant parts. We know that these conditions exist on the outside of the ambient environment, but how do the internal conditions actually then affect the plant's growth and reproduction? So firstly, a little bit more background information. We know that the atmospheric conditions within the plant parts are very little studied, and this includes the flowers, stems, fruits, and galls. Now, our project aims mostly to focus on these hollow structures of stems. We also know that transpiration has an effect on the temperature, the internal temperature of plants. So transpiration, of course, leads to the evaporation, which causes the cooling of plant tissue. And now there are also other mechanisms that are known, which are metabolic heating, which essentially the plant will increase the rate of cellular respiration under colder temperatures. And we see this in plants like the sacred lotus and skunk cabbage here in Ontario. But there are some special plants that are capable of metabolic heating as well. But in our project, we aim to understand the passive heating, which comes from the sun. So the mechanisms of microgreenhouse heating are very complex, and it's a very complex thermodynamic system that is held within a stem. So to explore this, we've broken it down into a series of steps. So firstly, the sun hits the wall of the plant, coming into the arrows A, and then it can be reflected through number 1, absorbed or transmitted. Now, the transmission is something we'll talk a little bit about later, as this depends on the translucency of the plant material, and this is something that we've been able to increase our methodology around and be able to quantify a little bit better after this year of fieldwork. So the absorbed radiation can then be conducted throughout the plant tissue. It can be lost into the lumen or into the airspace of the stem, or it can then be transmitted and conducted further. And this heat is adding energy to the lumen, which is the basis of our hypothesis of the microgreenhouse effect, of this elevated temperature within these hollow spaces of plants. And so this radiant energy can also be reflected and absorbed in a number of other ways. It can be absorbed by the atmosphere in the lumen and then be converted again through conduction, convection, or re-radiation. And all of these aspects contribute to the greenhouse effect within the stem. Heat in stems can then, once the heat is in the stem, it has a few different places it can go as well. So it can be liberated by radiation and conduction into the environment. So this can actually mean that the plants are influencing the ambient environment as well. It's not just vice versa. And that some heat can also be conducted away from the phloem and xylem through, and then through transpiration, that heat is going to leave the plant. And some is also used in photosynthesis and metabolism, which is not shown here as that happens on the cellular level. And this is a more of a schematic diagram published by Dr. Kevin in 2018. So this is the model that we have been working from, and we're essentially trying and getting closer all the time to measuring more and more of these processes within the stem. So to do this, we are using 2 model organisms. We are very lucky to have been able to continue some of our field work this summer at the pumpkin farm called Strom's Farm here in Guelph. And last winter, before the lockdown, we were able to complete work on Gerbera daisies at Van Geest Brothers greenhouse in Grimsby, Ontario. So yes, the names of these model organisms are Gerbera jamesoni or Gerbera daisies and Cucurbita pepo. The Cucurbita pepo that we were studying, they do have very numerous varieties. They have 25 varieties on their farm. So we were looking at the larger and smaller varieties. And for the Gerbera daisies, they have about 50 varieties in their greenhouse, all with a variable stem type between hollow and solid types. The hollow stems, they do cause problems for growers. They don't like it when they bend. And if you get a bouquet of Gerbera daisies, you certainly don't want them to fall over at your dinner table. So you want them to be able to be solid. So the greenhouse growers are interested from that perspective. And we know that the stem shape, it doesn't just influence the appearance of the flower, but it can also influence how fast that flower is able to grow and reproduce ultimately. So the temperature has a lot, we can learn a lot from our analysis of these flowers, and a lot from the pumpkins as well, as they have very large hollow stems in all of their varieties. So, well, here I'll explain some of the methods we are using to understand these mechanisms of heating within enclosed flowers, fruit, and stems. So on the right, you see a photo of a data logger that is inserted into the stem of a Formica plant. And this is one of the methods that we use to measure temperature. It's working with thermocouples in the field. And our lab, we have over the past 3 years, we have collected a lot of long-term data on the elevated temperatures. We have found temperatures up to 13 degrees warmer than the ambient air. These were from mostly from milkweed pods, have the most extreme results. And next, cucurbita, we found up to 10 degrees warmer, and in Gerbera stems up to 8 degrees warmer. So there is a degree of variation within our study plants, but we are still working to analyze the results from these plants and more, which include amaryllis, daffodil, physalis, and Japanese knotweed. So the shape and form measurements that we are taking in our lab, this has also been a major area of work. That's because as there have been no reviews of hollow stem plants based on geographic or environmental conditions. So it's really interesting to be able to collect these samples. So we have found, or we have taken samples of the physical dimensions of over 500 plant specimens that have now been cataloged and ID'd and are on file at the University of Guelph to go into the herbarium once everything opens up again. And as well as the shape and form measurements, this past summer, we have conducted a lot of growth rate measurements on our model organism to compare the sunny and shady sides of the field and how the growth rates are between those. And reflectance and absorbance spectrophotometry, this began in January 2020 in Amaryllis. We had to put this project on hold as we wanted to use some growth chambers on the University of Guelph, and this access was not available. So we decided to take some of this out into the field as well, which was a success that I'll talk about later. Histology methods, we were not able to use as much microscopy this year as we hoped, as of course the labs were closed. So the samples, we were not able to look at them under the microscope, especially not an electron microscope. However, we did make significant results with our IR thermography. So with the use of a thermal camera, we were able to develop a method to improve the accuracy of the surface temperature of plant surface temperature in horticultural applications. And this is presented in our paper by Byerly, Dr. Kevin, and Ali Badi, and myself from 2020. And another area of the research that we were not able to progress as far on this year because of COVID-19 was the thermal conductance of plants. We have purchased some stem flow meters, but these experiments are on hold due to COVID-19. So now I'm going to talk with you about our results and our methods for improving the accurate quantification of plant surface temperature. So our paper, Byerly et al. 2020, presents a method for the in situ calibration of an uncooled thermal camera. And with this method, we were able to reduce error and improve accuracies over observed surface temperatures of plant material. So our method, we used a FLIR E6 uncooled thermal camera. There is a difference between the cooled and uncooled thermal cameras of a significant number of dollars. So most people who are using thermal cameras will be using uncooled thermal cameras. So this method is very applicable to other people who are doing research and want to be able to take images inside and calibrate it at the location that they're working on. So our method, we used 5 varieties of Gerbera jamesoni. I mentioned before that their stem types are variable. So we wanted to choose varieties that would select more extreme ends of the spectrum of hollow and solid, and then somewhere in the middle so we can get a wider range of the actual stem surface temperatures to then use with the camera. And our data was collected, as I said, before lockdown last year, so January and February 2020, and we were able to analyze the results and get it published. So here on the right, we see the diagram of how we're actually doing the calibration in situ, where we have the thermal camera taking an image of the specimen, while at the same time we are recording the true temperature of the specimen by the use of a thermocouple and data logger, as we have shown in our previous methods. And this thermocouple and data logger were previously calibrated using an ice water and boiling water dew-point calibration. So we know the thermocouple to be calibrated. And then with this, we can then compare the thermal camera to the true temperature values. So our Gerbera varieties that we chose are shown here on the left. They are Prestige, Brunello, Panama, Rendezvous, and Toast. And now you can see that these represent, again, the spectrum between the darker shades of red and the paler shades of pink or white and yellow. And then we also chose varieties that have pale centers and darker centers. As one of the aspects of using a thermal camera, if you're familiar, is that you know there are constants that are set for the emissivity values of the specimens that you're looking at. You need to input the proper emissivity value that is based actually on the color and on the reflectance of the tissue. So this is why we chose a variety of different colors. On the right-hand side, you can see our image results from when we were taking thermocouple measurements and the FLIR at the same time, and the FLIR images at the same time. So you can see that the scale on the right hand shows that the purple colors are colder than the yellow or warmer colors. You can see that the flower is about well, it's hard to say here because I haven't actually done the analysis on this exact photo, but you can see that it is warmer than the surrounding stem and leaf tissues, and you can see where the thermocouple has been placed. Sorry. Okay, so now I'll explain how we use this image to actually figure out new calibration constant parameters that are intrinsic to the thermal camera. So we were able to derive these constants based on images that we were shown previously, and we were derived the new constants for the camera for both of the flowers and stems for each of the Gerbera daisy varieties we were looking at. And then next, the the bias and the root mean square error of the surface temperatures using the default manufacturer camera settings, and then our newly calibrated camera parameters were then calculated and compared. So here we can see our results that show linear calibration of the thermocouple versus the thermal camera of the FLIR E6. So on the right-hand side, you can see our graphs show the y-axis is the thermocouple temperature, and the x-axis is the heat of the object. And this is measured in AD count, which essentially refers to the pixel excitation in the camera. So the black triangles are the thermocouple temperature. Green squares were the thermocouple, or the image temperature for this, for each pixel that was calculated using the default camera parameters. And then the red circles are the calibrated parameters that we used based on the equations and that are intrinsic to the camera. So you can see that the red line, which represents the calibrated camera, is actually a lot closer to the thermocouple temperature than the default camera, where you can see, especially towards the higher end of the scale of temperature, there's a more drift of the camera to be overestimating the true temperature of the material. And we have shown the results for the Brunello and the Rendezvous flower, but we have repeated these for all of the different flowers. I just didn't want to bore you with all of them, but they show the same trend. And the significance of our results show that this linear relationship exists between the calibrated camera and the thermocouple temperatures more closely than with the default camera settings. Basically, researchers can use this method to improve the accuracy of their results.
There would be 5 minutes left.
Okay, thank you. So we have calibrated camera bias and error values were reduced by at least 89.1% between the default and the newly calculated error values. And like I said, most researchers have not used calibrated cameras, or they have calibrated elsewhere than their data collection sites, and the environmental conditions of the area that you're measuring, as well as the light conditions, it will change, and thus you can improve the accuracy of your results by calibrating in situ, how we have done. So our further studies, we actually were able to access a higher resolution camera. You can see the difference of resolution between these two images, and we're able to conduct longer term data collection, which are still in analysis. So here's a few more photos to share with you from that, which is from the nectaries of pumpkin flowers. They were about 3 degrees warmer than the surrounding air during the nectar production. And we also found that these pumpkin flowers have a humidity gradient starting after the flowers begin to open. So we're understanding this as it could be a potential pollinator cue for bees to come and visit the flowers before they close up. And the temperature and humidity gradients are related to this. We do have the next project that we were able to have good results with this year were the pumpkin stems, where we were looking at large versus small pumpkins, as well as sunny versus shady pots with 2 plants per pot and multiple stems per plant being studied. And we recorded the internal and ambient air temperature from June to August and collected daily plant growth data. Now we do have some results of the spectrometer, which are still in analysis, but you can see on the right-hand side that when the plant tissue was placed over the spectrometer, shown in this image here, and then the graph of the results are here, you can see that, of course, there's more green light passing through and less red light passing through, as that is what's used for the photosynthesis. So we want to compare how the light spectra inside the plant changes throughout the season and how that can also be reflected in the success rates of the plant. So these results are still in analysis. We have found that the stems reach about 10 degrees warmer than the ambient air temperature, and this difference occurs between 10 a.m. and 2 p.m. So this is providing evidence for our microgreenhouse effect. And just the one last point on the modeling of the microgreenhouse effect. So with all of these results that we have obtained from our model organisms, we aim to put them into a model where we can predict how a hollow stem's internal temperature may be affected by the ambient air temperature, solar radiation, and the plant tissue properties inherent in the plant. So these results I'm working on for my master's, and I hope that they will be useful to growers' environmental management plans of hollow stem plants, and I hope they will provide a theoretical basis for the effects of extreme weather conditions on hollow stem plants as well. So thank you very much. For our sponsors as well, again, to COHA and the AAFC.